Okay, let's begin the session with Kitab Al-Fatihah. Okay, all right. Sorry for having this class at night, but there's not much option with me. Okay, all right. Let's proceed.
Doctor, I'm supposed to share my screen, but I have unstable internet, so one of my colleagues will share the screen. Can you wait for a while? I'm very sorry.
Okay, it's all right, no problem. So how are you guys finding this ERTL, this forced online teachings? How are you guys coping?
Not sure. Maybe okay.
Not
maybe not.
Not sure. Why not sure? Do you think it has been going okay so far?
I think it's okay for me. But for those who have poor internet connection and so on, so they might not be so okay. Maybe.
Can I present now, Doctor?
Oh, okay, all right, all right.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I bid to Dr. Syed and everyone for attending our seminar today, which is entitled Genetics. Next slide. Okay, this is our table of contents, which will be presented by 4 presenters. Next. The first subtopic will be presented by me, Putri Sofia binti Azlan, metric number 1817520, on the molecular basics of genetics. Next. This is my outline. Next. Okay, first we'll start with the introduction. Next. Okay, we begin with the definition. So what is genetics, molecular biology, and molecular genetics? So genetics is the study of heredity and of the mechanisms by which these genetic factors are transmitted from one generation to the next. Molecular biology is the field of biology that studies the composition, the structure, and the interactions of cellular molecules. However, molecular genetics is the field of biology that studies the structure and function of these genes at a molecular level. Next. Okay, so inside of a human cell, we have a nucleus, and inside the nucleus, we have chromosomes. Chromosomes are fine thread-like structures which are made out of thousands of genes, and these genes are made out of strands, DNA strands. And in 2001, the Human Genome Project resulted in the first publication of the human genome sequence, and the human genome has approximately 25,000 genes. Next, we'll be talking about the central dogma of molecular biology, which was termed that way by Francis Crick in 1958. Which is basically the fundamental process of which heritable information is translated from genetic code to function in the living organism. So it's basically the process of which DNA is transcribed into RNA and RNA is translated to proteins. Next. Okay, next we begin with chromosomes. So chromosomes are fine thread-like structures of nucleic acids, DNA, and protein found in the nucleus of cells. It carries genetic information in the form of genes, as I've just mentioned. And in one chromosome consists of 2 identical chromatids, if you can see that picture there. And in the middle there is the centromere. Centromere is the constricted point of the chromosome. And in a chromosome, we have 2 arms. We have the P arm, which is the short arm. You can remember it by P for petite, and Q arm, which is the long arm structure. So these 2 identical chromatids are an exact copy of each other and contains one DNA molecule. And this DNA molecule is this long string. This DNA molecule is long string like molecule, which is formed into a compact. It's coiled around a protein called histones. Next. So the human chromosome all human cells has 46 chromosomes, which is equal to 23 pairs. So you can see that big green box, there is 23 pairs of autosome, and below there is one pair of sex chromosome. So two X chromosome for female and one X and one Y chromosome for male. So a copy of each chromosome is inherited from each parent. So one is maternal, one is paternal, and both of the chromosomes of each pair, they carry genes that control the same inherited characters. Next. So you can see that 46 chromosome, that little dotted line there, 46 chromosomes. If you were to unwind the supercoiled DNA, it will be about 2 meters long. And this 2 meter long string of DNA is actually found inside a nucleus. Can you imagine that? Inside the nucleus of a single cell. So that is how tightly coiled the chromosomes are. Next. So this is an example of a human karyotype of a male. Next. Next subtopic is genes and allele. Next, okay. For genes, genes are basically, if you can see that blue and pink genes there on the left side of the picture, it's basically a segment of DNA molecules that codes for a particular protein or trait. And these genes are found on a particular location of the chromosome called a locus. That is where you can see that band of pink and blue on the chromosome there. So it's basically, it codes for a particular protein. For allele, you can go to the right picture. The alleles, there are two types. They are recessive and dominant. So these allele, they are the genes that are found at the same locus on homologous chromosomes that control the same characteristic. So since gene is, it codes for a particular protein, allele, you can think of it like different variations of genes. So if you can see that in individual A, it's heterozygous because there are two types of allele, as I've said. So if you can see the blue band, which is allele for blue eyes, and the brown for allele for brown eyes. So if the human has two of these alleles of different type, they will be heterozygous. If individual B, which dominant for brown eyes, both of them are dominant, then what is manifested is brown eyes. For the heterozygous, what is manifested is the brown eyes for dominant. And individual C, the recessive type of allele will only be manifested when both of them are homozygous recessive. Next. Next, we'll talk about DNA. Next. So DNA, there are polymer of nucleotides which are conveniently linked by 3' to 5' phosphodiester bonds. So you can see that picture on the bottom left, the one with the circular dotted lines. DNA consists of 3 basic components, which is the circular one is the phosphate group, the pentose deoxyribose sugar, and after that the nitrogenous bases. You can see the one labeled in CGAT. So there are 4 different types of nitrogenous bases. A for adenine, C for cytosine, G for guanine, and T for thymine. So DNA, is a structure of a double helix and is anti-parallel to each other because the nucleotide they are connected in base pairs. So if you can see the most right picture, you can see that the base pairs for adenine, it will pair up with thymine. For guanine, it will pair up with cytosine. And the backbone of the DNA is the deoxyribose phosphate backbone. And the base pairs are connected to each other by hydrogen bonds. Next. So the function of DNA, it has 2 main functions. One is for replication and one is for transcription. Replication is basically so that it could provide the daughter cells with the genetic information that is possessed by the parents. And for transcription, DNA, it functions as the source of information for the synthesis of all protein molecules of the cell as well as the organism. Next. Okay, now I'll be presenting about DNA replication. So first of all, that circular purple thing on the left picture is the helicase. It will unwind the DNA strand into 2 single-stranded. So the yellow circular thing at the DNA strand is basically single-stranded binding protein. It actually stabilizes the unwound DNA strand. And then after that, okay. So the green part, okay, that is the primase. It was synthesized for RNA primer. And then after that, the DNA polymerase 3, it will actually synthesize the DNA in the 5' to 3' direction. And it will read the template strand from the 3' to 5' direction. So that is one thing to note. So this is called the leading strand. The left picture shows the leading strand, the one in the blue. For the right picture, you can see that there is another DNA polymerase and DNA ligase there. That will be the lagging strand. It's called the lagging strand because it is the DNA strand is synthesized in fragments. So these fragments are called Okazaki fragments. So the primase has to continuously synthesize RNA primer so that the DNA polymerase 3 will synthesize the DNA. And then after that, these Okazaki fragments will be joined together by ligase. So it will be the leading strand is called the leading strand because you can see that the top on the left, on the right picture, on the top one, the DNA polymerase, it synthesizes the DNA in the direction of the replication fork. So it's synthesized continuously. However, for the strand below there, it is synthesized in fragments. That's why it's called the lagging strand. Next. Okay, this is what I've explained before. Next. Okay, transcription. Transcription, as I said, is basically the process of which the information of the DNA strand is copied into a new molecule of the messenger mRNA. So what happens is, you can see that purple circular thing, which is the DNA RNA polymerase. It will actually bound to the promoter region of the DNA strand, and it will unwound the DNA into two single strand. So it will also read the DNA from a 3' to 5' direction, in which it will synthesize the messenger RNA in the 5' to 3' direction. So the template strand that it uses to synthesize the messenger RNA is in the 3' to 5' direction. So one thing to note that when it synthesizes mRNA, it will replace the thymine with uracil. So that is the process of initiation in which that it bounds the promoter region. And once it starts the elongation process, it will basically synthesize the messenger RNA complementary to its base pairs. And after it reaches the terminator, it will signal for a termination signal. And after that, the RNA transcript and the RNA polymerase will dissociate from the DNA strand. So it produce a new RNA transcript. Next. So this is basically what I've explained before. So that so the RNA polymerase, which is that big purple thing, it actually bounds the promoter region. That is called the initiation stage. And then after that, it synthesizes the complementary base pairs in the 5' to 3' direction. So it will continue to synthesize this messenger RNA called the elongation stage, and it will reach the termination stage, in which the terminator region, it will dissociate the RNA polymerase and the messenger mRNA from the DNA strand. And then after that, you can see that that long string red thing will actually be the new messenger RNA transcript. Next. Okay, so it's called the pre-mRNA because it's just been synthesized, and it has a 5' as well as a polyaethyl by polyadenylation. This happens because the 5' and the polyaethyl, it helps to stabilize the messenger RNA so that it doesn't be degraded from nucleases. And it also, this messenger RNA also it undergoes splicing, in which the introns are removed. Next. So the splicing is basically the process that it removes the introns and joins the exons in a primary transcript. So the red line is the exons, and the one that was removed was actually the introns, which is the yellow strand. So the exons, why it is joined together? Because these are the region that contains the code that ultimately corresponds to the sequence of amino acids, while the introns, which are spliced out, are the intervening sequence that do not become part of the amino acid sequence. And this reaction is catalyzed by spliceosomes. Next. So next process is translation, in which the process where the amino acid from the messenger RNA are decoded to produce a specific amino acid chain or polypeptide. But before we go to translation, I'd like to explain a bit more about codons. Can you go to the next 2 slides? Another one. Okay, codon. Codons, why you need to know codons before we go to translation is because it's the sequence of 3 DNA or RNA nucleotides that correspond to a specific amino acids or a stop signal during protein synthesis. So you can see that on the top left one, you can see that uracil, it's actually, it codes for a phenylalanine. So every codon, it codes for a specific amino acid. So every three nucleotide, it represents a single codon. Since there are 4 different nucleotides, and the codons are read in 3, so 4 to the power of 3 gives you 64 different combination of bases, and 3 of which you can see that I circled in red there, 3 of which are stop codons. And one thing to note that AUG, the one encircled in green, is methionine, is an amino acid, and the AUG codes for the start codon. Okay, can you go back to the previous translation? Previous. Okay, so you can see on the left, the furthest left, that is the translation apparatus in which that the large and small ribosomal unit, as well as the messenger RNA and the tRNA, which is connected with an amino acid, methionine, it binds to the messenger RNA at the start codon, AUG. So it codes for methionine, which is the start codon. That is the initiation stage. The next picture shows you how a methionine is attached to proline amino acid. You can see that methionine, a circle blue thing, will actually connect with the proline. This is because the next tRNA, it brings the amino acid as it codes for. So basically the codons, it codes for a specific amino acid. So it brings the amino acid and it will bind together. So this process continues on as the elongation stage, and it produces a long polypeptide or a specific chain of amino acid. So the ribosomal unit will actually move to the next codon, and then it moves to the next codon until it reaches the end, which is the stop codon. So this stop codon signals it for the translation apparatus to dissociate. Thus, it produces a new polypeptide chain. You produce a polypeptide and a new amino acid chain, or a new amino acid chain. Okay, next. This is basically what I've just explained. So what happens at the end there? There's a completed polypeptide, and then after that, the translational component is recycled so that the process could be undergo through again. Okay, next. Next. Okay, now we talk about mutation. Next. Mutation are, I'm sorry for numbers.
Before you go further, what's the latest thing, latest hype about mRNAs?
Latest hype.
mRNA has been in the news for the past 1 week.
The vaccine, Doctor.
Oh yes, both the Moderna and also Pfizer vaccine. Malaysia dah beli, Malaysia dah bayar for the Pfizer vaccine. Tinggal nak tunggu tinggal nak tunggu FDA approve je. Once FDA approve, dia akan start bagi, and the first ones that's gonna get these vaccines are the healthcare workers. So us lah among them. So both technology, Moderna and also Pfizer, both uses mRNA. So mRNA usage of mRNA vaccine is a new technology. It's a very new technology. So the mechanism of how it acts is like the presenter presented just now. So previously, how does vaccine? So vaccine sama ada, it's either active or passive kan? Passive, active or passive immunity. Passive immunity ni macam you bagi antibody, macam untuk RSV punya vaksin kan? You bagi monoclonal antibody. So that is passive immunity, ataupun you bagi hepatitis B immunoglobulin. So those are passive immunity. You bagi immunoglobulin, you bagi antibody tu. Those are passive immunity. The active one are, you can either give live, you can give kill, you can give attenuated, you can give polysaccharide, you can give conjugated. So there's so many types of vaccine. But what happens is, in active vaccination, active immunity is where you introduce to the body an antigen. Maknanya sama ada daripada virus tu sendiri killed ataupun live attenuated ke just polysaccharide vaccine or conjugated vaccine dan sebagainya. So you bagi just a part of the antigen. So your body will detect this antigen and your body will produce antibodies against that antigen. So this is how vaccination works traditionally. So now the thing that is coming now is mRNA vaccine. So where this vaccine will teach the body to produce protein. Antibodies are protein. Immunoglobulin are protein. So instead of giving antigen, we are giving mRNA. And mRNA does not actually affect the DNA or anything. And so it's basically just protein. So you translated it into a protein, production of protein, which is antibody, which is much safer rather than the other old methods of vaccination. So this is a very new method of vaccination. The idea is not new. The idea is old. The idea of using mRNA, using genetic and so on and so forth was a very long time ago. But I think COVID-19 vaccine will be the first mRNA vaccine if it's successful. We'll see how it goes. So mRNA is a very interesting. It is the thing for the past 1 week. Okay, proceed.
Okay, thank you, Doctor, for the info. Sorry for my lack of knowledge. Next, we proceed with mutation. So mutations are permanent changes in the sequence of DNA, and it can affect a single nucleotide pair or larger genes of a chromosome, and it may cause severe or permanent disability that affects the quality of life of an individual. And mutation can be caused by a lot of reasons, such as environmental factors, which are chemicals, radiation, or UV light. And it could also be due to error made during meiosis and mitosis, which could lead to replication error. And the classification of mutation, there are genome mutation, chromosomal mutation, and gene mutation. Next. So for genome mutation, what happens is that there's a loss or gain of a whole chromosome, or it could cause monosomy or trisomy. So we know that Down syndrome is trisomy 21. So basically there's an addition of a chromosome in chromosome 21. And for Turner syndrome, there is a loss of, in Turner syndrome is monosomy X. So you can see there's only one X chromosome. So the person will suffer from Turner syndrome. Next. Next is chromosomal mutation, which is the rearrangement of genetic material, which causes visible structural changes in the chromosome. So it has a in chromosomal mutation, there are different types. There are deletion, duplication, inversion, and translocation. So it just as its name suggests that deletion is a removal of a chromosomal segment. So you can see B will actually be removed, and that's called deletion. For duplication, there's a repetition of a segment, so B will be duplicated. So for inversion, there is a reverse of a segment within a chromosome. So ABC, it will become AED. There's an inverse within a chromosome. However, translocation is the movement of a segment from one chromosome to another non-homologous chromosome. So instead of DEF, it will actually translocate to another non-homologous chromosome. Okay, next. The last classification is gene mutation, in which there are different subtypes. So the first one is point mutation, which is a single base substitution. So what happens is that a single nucleotide is substituted by a different base, which causes the replacement of an amino acid by another in the protein. Because remember, codon, it codes for a particular protein. So once you change the nucleotide, it can actually replace the whole amino acid in the protein product. So you can see the picture on the right. The point mutation has different types. There is silent, there's nonsense, and there's missense. In silent, you can use the no mutation as a reference.
Bukan nonsense, nonsense.
Nonsense.
Bukan nonsense ni macam nonsense you're talking nonsense, talking mengarut nonsense. Nonsense and missense.
Okay, thank you, doctor. I apologize. Nonsense and missense. So in silent point mutation, what happens is that the mutation occurs, but there is no observable effect on the phenotype, because the codons, there are lots of different combination of codons. There's a lot of combination which codes for the same. There are lots of codons which codes for the same amino acid. So for example, for the silent one, it's silent because the change of the nucleotide causes it to become the same. The change causes it to have the same amino acid as the one with no mutation. For the nonsense, the mutation that occurs, it causes the codon that codes for a particular amino acid to change to a stop codon. So there is a premature termination. So that will happen. So for the last one is missense. in which there are two types. One is conservative and one is non-conservative. Conservative means that missense mutation means that the mutation that changes the codon for one amino acid to another. The non-conservative is a radical change, while the conservative is not much of a radical change. An example of point mutation is that in sickle cell anemia, the GAG codon is changed to GUG. So instead of glutamic acid, it will code for valine. So it causes a mutant protein. Okay, next. Next is frame shift mutation, which is a change in every amino acid that follows the point of mutation. So an insertion and deletion of nucleotides can actually shift the reading frame of the DNA strand. You can see the middle picture, it's inserted a guanine inside the DNA strand. So this codon, it actually, since it's coded into 3, it actually changes the whole amino acid that follows the point of mutation. An example of frame shift mutation is in cystic fibrosis, there is a deletion of 3 nucleotides. The CTT, the one in the box below there, it is deleted from the coding region of the gene, which causes it to lose the phenylalanine in the 508th position. So this causes the person, the individual, to suffer from cystic fibrosis. Next. Next is a trinucleotide repeat expansion. Just as its name suggests, tri means there is 3 nucleotides in a sequence that is amplified. For example, in Huntington's disease, you can see the DNA strand below, CAG is actually repeated. So it will the amino acid that it codes for, glutamine, is actually amplified. So this causes the person to suffer from Huntington's disease. Next. So the last one is the importance of molecular genetics. Why you have to learn about molecular genetics. Next. What I found is that the importance of learning molecular genetics, it gives us an insight to heredity, genetic variation, and mutation, because genetic itself is the study of heredity. It also helps us for the diagnosis and management of genetic disorders. And lastly, for identification of genetic disease or carriers for genetic counseling. And just as Dr. has stated that the new vaccine that is just been released, and that is also one of the reasons why you have to learn about molecular genetics. Next. This is my references. Next. And that's all from me. Thank you.
Any question? Okay lah, clear lah kan. I think that's quite clear. Because semua orang tidur belaka dah. I assume yang tak ada kamera ni mungkin tidur dah kot. Ke apa? Okay. Alright, next. We have 3 more.
I'll share.
Bismillahirrahmanirrahim. I'll be the next presenter.
Sorry.
Isa, I'll present next. I'll share my screen.
Okay, jangan bubut. Okay.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh. So today I'll continue the presentation. Not today, tonight lah. Tonight I'll continue the presentation with autosomal disorder. So this would be my outline for my presentation, starting with classifications of autosomal disorders, the definition, and then continuing with the brief, not quite brief explanation on the autosomal dominant and also autosomal recessive diseases. So autosomal disorders fall under the categories of Mendelian inheritance, which includes the main 3 disorder, which is autosomal dominant inheritance, autosomal recessive inheritance, and also X-linked inheritance. So what is Mendelian inheritance? Mendelian inheritance, as described by Gregor Mendel himself in 1866 would be the transmission of inherited traits or diseases caused by variation in a single gene in a characteristic pattern. So to start, autosomal dominant would be the most common mode of Mendelian inheritance. Autosomal dominant conditions are caused by the alterations in only one copy of a gene pair. For example, the condition occurs in the heterozygous state, despite the presence of an intact copy of the relevant gene. For example, if I have two fingers over here, so if one of the fingers is affected or mutated, then the whole body would start to manifest the disease. So the condition occurs in the heterozygous state. So autosomal dominant genes are located on the autosomes. So like what Putri Sophia has presented just now, we have 23 chromosomes, our paternal and also maternal chromosomes. But for autosomal dominant, autosomal dominant diseases, it is only affecting the autosomes or non-sex chromosome. So males and females are equally affected. Each child from an infected parent has a 50% chance of inheriting the abnormal gene if one of the parents are having the disease or the mutation. So for autosomal recessive inheritance, it involves the mutation in both copies of a gene, which means both of my fingers are affected. So an affected individual is homozygous for the abnormal gene, having inherited an abnormal allele from each parent. So from here you can see that the father is a carrier, the mother is also a carrier, and the affected son or daughter would be having both of the mutated gene. Autosomal recessive is also affecting only the non-sex chromosomes, same as autosomal dominant. So autosomal dominant, it seemed to be quite straightforward, but there are some complicating factors which include the following, starting with variation. Variation to explain variation within a family, some affected individuals may manifest the disorder mildly, and others can manifest the disease more severely. So this may be the result of variation at other genes, or it can be due to environmental effects or just by chance. And then next for non-penetrance or penetrating factor. So penetrance or non-penetrance over here refers to the lack of clinical signs and symptoms in an individual who has inherited the abnormal gene, which means for certain diseases, even though even though the person is having the mutated gene, but not necessarily he or she will manifest the disease. And then for no family history of the disorder, it means that a new mutation in one of the gametes that leads to the conception of the affected person. This is the most common reason for absence of a family history in dominant disorders. For example, 80% or more than 80% of individuals with achondroplasia have unaffected parents, which I'll explain later. So it can also be due to parental mosaicism. It is very occasionally a healthy parent can harbor the mutation only in some of their cells. For example, a healthy parent only having the mutation in their gonads. Therefore, when the sperm and the ovum unites or fertilized, the affected gametes would carry the mutated genes. Then only causing the child to inherit the mutation. It can also be due to non-paternity. For example, a patient coming with a autosomal dominant disease, but the father or mother is clear. It can be that he or she is not the biological father. Then for homozygosity, it's in the rare situation where both parents are affected by the same autosomal dominant disorder, there is a 1/4 risk that a child will be homozygous for the altered gene. And this usually would cause a more severe phenotype, which can be lethal, as for example, with achondroplasia. Okay, so this is the example of non-penetrance. So we can see in the first generation, the father is affected with autosclerosis. But then, even though this is an autosomal dominance, we didn't see any child bearing the disease. But then on the third generation, suddenly the daughter of this son is manifesting the disease, which means it can be that it is non-penetrant in the second generation. It is quite rare for autosclerosis to have not a new mutation. Okay. So next, this would be the diseases, examples of diseases for autosomal dominance. And I will only be explaining on 3 of them: neurofibromatosis, achondroplasia, and also hereditary spherocytosis. For the other diseases, you can read it on your own, because it is just too much and too lengthy if I would like to explain everything. So first one would be neurofibromatosis. Neurofibromatosis are autosomal dominant disorders that causes tumors to grow on nerves, and it also results in other abnormalities such as skin changes, bone deformities, and others. So this affect 1 in 3,000 live births. It is also a highly penetrant condition, and 1/3 of patients who have new mutations, like what I've mentioned, which means neither of the parent are having the disease. It suddenly appear sporadically. And then it is believed that there are 2 types of neurofibromatosis, type 1 and type 2, but it is recognized that they are clinically and also genetically distinct diseases and should be considered separate entities. And tonight, I'll be explaining only on neurofibromatosis type 1. So this would be the pathophysiology of neurofibromatosis. The NF1 gene, which is the mutated gene, is the gene that is responsible to produce neurofibromin. So neurofibromin is a tumor suppressor, which is expressed in many cells, primarily in neurons, glial cells, Schwann cells, and also early in melanocytes development. So this protein, the neurofibromin, is a regulator of Ras guanosine triphosphatase activity, or this GTPase activating protein, GAP, GTPase activating protein. So it serves as a regulator of signals for cell proliferation and also differentiation. So the loss of function of neurofibromin will therefore remove this regulation and lead to uncontrolled cell proliferation. Neurofibromas and melanocytes in Café-Olé macules have a mutation in both NF1 allele. That's why patients with neurofibromatosis would have these Café-Olé macules, as for the mutation would mainly occur in the melanocytes, these glial cells, Schwann cells. So the mutation in melanocyte would manifest as Café-Olé macules, in the glial and Schwann cells as neurofibromas, the tumor itself. So how can we diagnose a patient to have neurofibromatosis type 1 would be if the patient is having 6 or more Café-Olé spots, which is greater than 5 millimeter in size before puberty, and also it increases in size after puberty, and more than 1 neurofibroma, which is the tumor, and axillary freckles, dark spots, brown spots at the axilla, and then optic glioma, which is a tumor at the optic nerve, and then leash nodule, which is a hematoma of the iris. Bony lesions can be sphenoid wing dysplasia, which can cause eye protrusion, and also if the patients have a first degree relative with NF1. So this would be the clinical manifestation. This one is the freckles. This is the Café-Olé spots, or we Malay would recognize this manifestation as a birthmark or tanda lahir. And then these leash nodules, the hematoma on the iris, lump on skin. It can also be under the skin. So, okay, this would be the on a real human being. So this is one of the severe case of neurofibromatosis. The tumors are all over the body. This is the axillary freckles, the optic glioma, the cafe-au-lait spots, and also the lesion nodules. So next disease would be the achondroplasia. Achondroplasia is it coming from the name. A means not having or tak ada. Chondro, cartilage. Plasia is growth. So not having the cartilaginous growth. Okay. So this disease is due to the mutation at chromosome 4, which results in impairment of the FGFR3 protein. So the protein is responsible for the production of the FGFR3 receptor, which I will explain after this, which inhibits the multiplication of chondrocytes. So longitudinal bone growth is driven by the proliferation and differentiation of chondrocytes in the growth plate of the bone. So the disease that caused the FGFR3 receptor to be overexpressed and overstimulated, and therefore would inhibit the bone elongation. That's why patients with achondroplasia would have the limbs shortened, large head, frontal bossing, and also depression of the nasal bridge. The hands are short and broad, and also a marked lumbar lordosis would develop. Lumbar lordosis is a curved lumbar, a severely or extremely curved lumbar. And sometimes patient can also present with hydrocephalus. So right here, you can see, so this is the FGFR3 gene, fibroblast growth factor receptor, which encodes for the FGFR3 protein over here. So this protein, this receptor would inhibit the differentiation and also proliferation of chondrocytes if it is bound by this fibroblast growth factor. But due to the mutation of the FGFR3, it causes the protein to be overexpressed and overstimulated. Therefore, if it's overstimulated, then the bone would continually be inhibited of growth. So that's generally how achondroplasia affects all patients. That's why the patient would be stunted. So these are examples of child patients. We can see here the lumbar lordosis. The patients are all having large heads, white fingers, and also short limbs. So this is how we differentiate between disproportionate dwarfism and also proportionate dwarfism. So it is two different entities. For proportionate dwarfism, it can be due to growth hormone deficiency, primordial dwarfism, and Séquel syndrome. What I'm trying to say is that proportionate dwarfism means every parts of the body is proportionate to each other. While for disproportionate dwarfism, we can see that patients having large head, short arms, it is disproportionate lah. If a person is short, then the head wouldn't be big, for example. So achondroplasia is the most common for disproportionate dwarfism. So next would be hereditary spherocytosis. So this disease occurs in 1 in 5,000 births in Caucasians or in whites. So it is one of the most common cause of hemolysis and also hemolytic anemia with prevalence, I've mentioned, 1 in 5,000 births. So usually it has an autosomal dominant inheritance, but in 25% of the patients, there is no family history and it is caused by new mutation. But we have to know that spherocytosis can also be due to autosomal recessive, and it can also be due to X-linked inheritance. But today, I would only touch on the autosomal dominant part of this HS. So it's caused by mutation in genes for protein of the red cell membrane, mainly spectrin and chirin or BAN3. So the pathophysiology of spherocytosis is involving 5 different proteins, but these three, spectrin and chirin and band3, are the ones that is common for autosomal dominant HS. The other two proteins would be beta spectrin and also protein 4.2. I'll explain later. Okay, so the manifestation of patients with HS would be, the first one can be jaundice. Patients with HS may come with intermittent jaundice due to the hemolytic crisis that occurs throughout their life. Patient may also come with severe hemolytic jaundice at their early life, which would need a proper care. And then for any patient can also comes with mild anemia, which usually present in childhood, but it can be increased in severity, especially when patients having infection. This is because some infection would cause the hemolysis or hemolytic crisis to increase in severity. Patients can also have splenomegaly due to increase in the blood sequestration and also filtration. This is due to the RBC elimination or the abnormal RBC elimination in the spleen. So when the spleen is overworked, that's why it enlarged. Then due to the hemolysis, it releases too much of bilirubin, and increase in bilirubin secretion would increase the chance of patients to be getting bilirubin gallstones, which is found in approximately 50% of patients with HS, and frequently are present in patients with very mild disease. Therefore, a periodic ultrasonic evaluation of the gallbladder should be performed regularly. Okay, so over here we can see a normal erythrocyte, normal red blood cell, and this would be the abnormal one, which is the spherocytes. So this is the protein I've been saying about, the band3 protein, the band 4.2, and the N-chirin protein. This is a spectrin alpha and also beta spectrin. So a deficiency in spectrin, like over here, this is the normal one, this is the abnormal one. So a deficiency in spectrin, band3 or N-chirin, results in the uncoupling in the vertical interactions of the lipid bilayer skeleton, and it would lead to subsequent release of membrane microvesicles. It is normal for any RBC to release microvesicles if the RBC is having problems in itself. For example, due to reactive oxygen species in the red blood cell, then it will release microvesicle. But in patients with hereditary spherocytosis, the microvesicle release is increased. So the loss of the membrane surface area would, without the proportional loss of cell volume, would cause the sparing of the RBCs. So when the red blood cells are abnormal in shape and size, then the spleen would recognize it as asing lah, red blood cell, asing. Okay, what do you say asing? Me, asing, you asing apa asing?
Foreign.
Oh yes, okay.
Alien.
Foreign.
Alien.
Foreign red blood cell. Okay, so that's why the splenic macrophage would engulf the RBC, causing increase in hemolysis. Okay. So this is the blood investigation. You can see this one is the normal red blood cell with central pallor, and this one is the spherocytosis, which is round in shape without having the central pallor. And patients with HS can have spherocytosis, spherocytes, and also polychromasia. Polychromasia is, by definition, is the difference in the chrome or the color of the RBC, and this is due to the new RBC that is being formed from the bone marrow. So an increase in the hemolysis would increase in the production of new red blood cell, and new red blood cell will be having different color than a matured red blood cell. Okay, so moving on to the next part, which is autosomal recessive. So this would be the diseases, some example of the diseases, which is sickle cell disease, cystic fibrosis, thalassemia, galactosemia, and others. And I will only touch on these two, sickle cell and also cystic fibrosis. So sickle cell disease is the commonest genetic disorder in children in many European countries, including the UK, which is prevalent in 1 in 1,000 live births. Sickle cell disease is a collective name. So it is given to hemoglobinopathies in which the HbS or the sickle hemoglobin is inherited. While sickle cell anemia occurs when both the beta globin alleles have the sickle cell mutation, which means if a patient having both of their parent with both of their parent as sickle cell anemia carrier, and the patient inherited both mutated gene, then he or she will have sickle cell anemia. But if only one of the gene is inherited, then it is called sickle cell disease. So this is the pathophysiology of sickle cell anemia. So in the red blood cells, the hemoglobin molecule has a highly specified conformation, allowing for the transport of oxygen in the body. And the absence of the globin chain mutation, hemoglobin molecules do not interact with one another, like this one. It is usually not interacting with one another. But in the absence, but in the presence of globin chain mutation, it results in the change in the conformational state of the hemoglobin tetramer, and therefore in the deoxygenated state of the RBC, the HbS molecules would now interact with each other. Then it will form the rigid polymers that give the red blood cell its characteristic sickle shape, like this one. So the lung is the only organ that's capable of reversing the polymers, and any disease of the lung can be expected to compromise the degree of reversibility. Usually for any red blood cell that have the HbS mutation, it can return to its original state. But after some times or after frequent sickling, it may be permanently sickled. And a reversibly sickled cell have a reduced lifespan, and it may be trapped in the microcirculation, which result in blood vessel occlusion, this vaso-occlusion. And therefore, it can cause ischemia in any organs or even bones. And this disease is exacerbated by low oxygen tension, dehydration, and also cool temperature. Why? Because low oxygen tension would increase the number of deoxygenated red blood cells, while dehydration and cold cold specifically can cause vasoconstriction, and when vasoconstriction is present, then the vaso-occlusion can be more severe. So when a normal red blood cell hemolyze, it will release its own content, and for a sickle cell red blood cell, it would release arginase and also HbS coupled with the iron 2+ ion. And this iron 2+ ion is regarded as a reactive oxygen species, and therefore would normally react with nitric oxide. But due to the increase in hemolytic crisis, then more nitric oxide is needed to neutralize the ferrous ion to convert it to the 3+ ion. And if the nitric oxide is reduced in its bioavailability, then as we all know, the function of nitric oxide is to vasodilate. If NO is reduced, then less vasodilation can occur. Therefore, increasing the severity of the vaso-occlusion. And for normal hemoglobin, normal RBC, they would release hemine, this iron compound. And this iron compound would would trigger inflammation by activating neutrophils and also the epithelial cell. But in sickle cell hemoglobin, it is believed that the hemine is increased by 3 to 5-fold. Therefore, more inflammation would result. Okay. So next would be cystic fibrosis. It is the commonest live limiting autosomal recessive condition in Caucasians, with incidence of 1 in 2,500 live births, and also carrier state of 1 in 25. Can you believe that in every 25 person, one of them would be the carrier of cystic fibrosis? That is how common this disease is. So it is a multi-system disorder of children and adults. It causes this dysfunction of the cystic fibrosis transmembrane conduction regulator protein, or CFTR. So the CFTR protein is a 1,480 amino acids protein. It is expressed largely in epithelial cells, especially in our airways and also in our GIT, the sweat glands, and also the genitourinary system. So it functions as a chloride channel and other regulatory functions. So this poster is quite a holistic explanation on the pathophysiology of cystic fibrosis. So we can see the mutation of cystic fibrosis CFTR gene on chromosome 7 leads to the dysfunction of the CFTR protein itself. So this would lead to chloride channel no longer allowing for chloride transport. So in sweat glands, the CFTR proteins are responsible for the reabsorption of chloride ion. In duct of epithelial tissue of other parts of the body, it facilitates diffusion of chloride ion into secretion. So therefore, if patients with cystic fibrosis would be having less reabsorption of chloride ions in the sweat, therefore increasing the secretion of chloride ions into sweat. And this is the diagnostic criteria for cystic fibrosis, which is increasing chloride concentration in sweat of the patient. And this one, if less chloride diffusion into periciliary fluid, then less water composition of periciliary fluid, which means we all know that water follows the movement of salt. And if less chloride ion is diffused into the fluid, then the less water is also diffused into the fluid, and it would lead to a thicker mucus secretion. So if it is thicker, then less clearance of the secretion. Therefore, it would accumulate, which will lead to obstruction. So in GI tract, it reduces the movement of intestinal content. So it can lead to this allele obstruction syndrome. In newborns, it can also increase the retention of meconium, increase the reabsorption of bilirubin, and it can prolong jaundice in neonates, which is also one of the manifestation of cystic fibrosis in neonatals. So this is the other ones in upper respiratory tract, in pancreas, in lower respiratory tract. So the manifestation of cystic fibrosis is very systemic, very general. It can happen in every part of our body. So over here in the lower respiratory tract, chronic productive cough due to the thick mucus production and less clearance of the secretion itself. So you can read everything else. I guess that's all from me.
Any
This
question?
would be the references.
Any question to the presenter? Okay. Autosomal dominant, autosomal recessive. These are favorite questions in exam. But simple questions lah. Usually OSCE kan. Kita bagi example of family tree, and then we'll ask you, what is this type of inheritance? And list 3 common diseases with this inheritance. So kita tahulah example of common autosomal dominant, example of common autosomal recessive. Okay. Any question?
Just one question for clarification. When you said that reduced chloride diffusion in mucus secretion, meaning that it makes the mucus more thick. Is that true?
Yes, yes.
My understanding. Oh, okay.
That's true.
Okay, thank you. Just for clarification only.
Okay, thank you so much.
Alright, let's proceed with the next presentation.
Apa Nana ni? Nanti apa?
Okay, Bismillahirrahmanirrahim. My name is Nuriza Zahira binti Zamri, and today I will be presenting regarding the sex-linked disorder. Okay, first of all, we start with the sex chromosome. So before this, Putri has explained what is chromosome and what is contained inside the chromosome, and what does a specific gene does if it was affected. So basically, sex chromosome is the 23rd pair of human chromosomes. There are two types of sex chromosome, which is the X chromosomes and the Y chromosomes. For female, they will have the both XX chromosomes, and for male, they will have the XY chromosomes. As you can see from the picture, that the X chromosome is slightly larger from the Y chromosome. So in relation of the size, of course, the X chromosome will have more genes as compared to the Y chromosomes. Okay, the next one is regarding the sex-linked patterns of inheritance. The first one is X-linked inheritance. So X-linked inheritance can be divided into X-linked dominant or X-linked recessive. So the first one is X-linked dominant. So when saying the X-linked dominant, the trait will manifest in patients that have the X big H or X big H small h, or in male patient that have the dominant X. Okay, so the gene controlling the trait is located on one of the chromosomes is expressed, so only one gene is needed, and therefore there is no carrier status. The trait will appear in every generation. For daughters of affected father are all affected, since affected father must give the X chromosomes to the daughter. So if the X chromosome of the father is affected, then therefore the daughter will get the disease or the trait. And the sons of affected father are unaffected because they will receive the Y chromosome from the father, and the X chromosome that is the dominant trait will not be given to the son. For homozygous mother, which have the both X big H, the trait will be manifested in 100% of the child with each pregnancy. For heterozygous female, the risk is about 50%. So this can be explained with this image here. As you can see on the left side, where the affected mother has the heterozygous X chromosome, you can see that the chance of the child getting the trait is about 50% for both sons and daughter. As you can, for the right side, you can see the affected father has one X dominant gene. So the chance of the son to get the trait is 0%, while the daughter will always get the trait from the father. The next one is X-linked recessive. So X-linked recessive can be shown by the type of X small h or X small h or Y. This trait will only be expressed if they are not other dominant gene. So if both are recessive, only then the trait will accept. So the transmission of the traits will occur from fathers to all daughters, since again the fathers will give the X gene, and affected mother have a 50% risk of transmitting the gene to her friends. So maybe if we see this image, you can see that if the mother has the heterozygous X genes, the son will get the X small h will be affected, while there is also 25% there will be a carrier for the daughter, since the daughter also have another one that is X chromosome that is normal. So for the right one, as you can see, the father has the recessive X gene. So this will result in all carriers for the daughter and unaffected sons, because they only receive the Y gene from the father. The last one is Y-linked inheritance. So Y-linked inheritance is associated with male, since normal female doesn't have Y chromosomes in their body. So only male are affected. And mutated alleles from the Y chromosomes are always passed on from the father to the son. but the father, of course, can transmit these genes to his daughters. So next one is the sex linked disorders. I will just explain briefly regarding these disorders. This dominant disorder I put in is the X-linked hypophosphatemia, incontinentia pigmenti, fragile X syndrome, and also Rett syndrome. The first one is X-linked hypophosphatemia, or sometimes they is all called the hypophosphatemic rickets. So the major complications of this hypophosphatemia is actually the rickets. So that's why they call it sometimes hypophosphatemic rickets. So the XLH is a systemic disorder. The mutations come from the regulating genes homologous to endopeptidase on the X chromosome, which is the chromosome X. And the patients will have significant phosphaturia. So as patients ingested or have milk with a lot of phosphate, and it will be converted into phosphorus in the kidney. But since the presence of mutation of this gene, the phosphate cannot be converted to phosphorus. So it results in a lot of the phosphate being wasted in the urine. So we call it phosphate wasting. So phosphate is very important in the bone development. We can see that in patients who have X-linked hypophosphatemia, the child will have, of course, a slow growth rate, and the patients will become reluctant to bear weight when beginning to stand or walk. So you can also see in the picture that they have sort of malleable bone, is that word? Okay. And older children may also experience multiple dental abscesses, as I shown before. Other features include the dentition, deafness, cardiomorphation, extraskeletal calcification of tendons and ligaments. This is all because of the lack of phosphates. Okay, the next one. This is the summary of the symptoms that can be manifested. The X-linked hypophosphatemia may not be manifested in pediatric, but can also develop after several years and then manifest in adult. Okay, the next one is incontinentia pigmenti. So incontinentia pigmenti is basically a disturbance in skin pigmentations, sometimes associated with malformation of eyes, skeleton, teeth, and other systems. So the picture on the left is actually the vesicular rash with eosinophilia. And the picture on the right shows that the black quite hyperpigmentation. So you can see like a vertical line there that usually found in the adolescents as well. And you can also have the linear atrophic Hallis lesions. Sometimes patients might be patients who have incontinentia pigmenti might be afraid that they will lose some hair as they grow up. So yeah, that's all. The next one is the fragile X syndrome. The fragile X syndrome, the common physical features is actually broad jaw, large ear, and long face. So fragile X syndrome, or called Martin-Bell syndrome, is caused by the FMR1 gene, the mutation in FMR1 gene that causes the slowed brain development, because there are certain proteins that are essential for brain development that cannot be made. So for developmental history, the patients will have the developmental muscle delayed, especially in gross motor. In cognitive future, they may have mild or severe mental irritations. It depends on the severity of the disease. And for the term of neuropsychological, they can have depression, general and superficial anxiety, etc. For musculoskeletal, they may have a pes planus or pectus excavatum. So pes planus is actually flat foot, and pectus excavatum is actually sunken chest or sunken breast. They can also have scoliosis. But what can be seen in this patient is also feeding and toileting difficulties. The last one for X-linked dominant is actually Rett syndrome. So the Rett syndrome is the neurodevelopmental disorder that typically is a degenerative, and it's caused by various mutations on the MECP2 genes. So for the Rett syndrome, the major hallmark for Rett syndrome is actually wringing of the hands or stereotypic moving of the hands. I could explain it on video. If you find some video that for patients who have Rett syndrome, they will have some repeated hand movements that they do it repeatedly and every single day. So for stage 1, they may have only developmental areas for 6 to 18 months, and then they will proceed for stage. If they proceed stage 2, they will have rapid deterioration for regressions. I heard some patients that actually neonatal baby began to speak like mama or dada, but then after just like after progressing for months, and then suddenly they stop speaking. So they will have these regressions. They will have the abnormality to talk, and then they proceed with stage 3 or pseudostrategy. Last, they proceed with stage 4 with late motor deteriorations. So in late motor deteriorations, you can see that they might not be able to walk or support themselves. Okay. The X-linked recessive disorder. So X-linked recessive disorder, the most common X-linked recessive disorder, I think so, is red green colours blindness. Second one is hemophilia, and then G6PD deficiency, and last is Duchenne muscular dystrophy. So for red green colour blindness, there are several terms for red green colour blindness. So the red green colour blindness are affected due to the photopigments of green or red cones of the eyes. So if the term is anomaly, such as deuteral anomaly or proto anomaly, so the affected patients may have mild symptoms if the patients have protonomia or absence of cone cells. They will have a severe color blindness, more severe color blindness. Okay. So you can see from the picture on both right and left that if the patients have deuteronomy, yellow and green will look red. It is usually mild and doesn't usually get in the way of activities. For protonomy, orange and red and yellow look greener. Colors are less bright. And for protanopia, there will be no working red cells. So red will look dark gray. And for deuteranopia, there will be no working green cone cells. So the red may look brownish yellow and green may look beige. So for tritanopia is not considered as part of X-linked recessive disorder. So I will explain. So the next one is hemophilia. So hemophilia can be divided into hemophilia A and hemophilia B. So if patients have hemophilia A, the patients will have deficiency in clotting factor 8, and if the patients have hemophilia B, the patients will have deficiency in coagulation factor 9. So to diagnose the patients with hemophilia, we can do the either the FPTT or PT test, or also we can do the specific factor of factor 9, and the level of factor 8 activity of factor. The patients may have mild diseases such as bruising, such as bleeding that does not stop when there are mild injury or easy bruising. But the patients can also have severe manifestations such as spontaneous hemorrhage. So the main treatment for this hemophilia and hemophilia B is to give the factor concentrate of factor 8 and or factor 9, depends on the severity of the patient. Okay. The next one is glucose 6-phosphate dehydrogenase deficiency or G6PD deficiency. This is the most common enzyme deficiency. Most patients doesn't really have the clinical manifestation, doesn't show manifestation unless they are affected with oxidative stress. But the patients may have the history of neonatal jaundice, and this manifestation, the patients may be triggered for intravascular hemolysis if they are triggered by something such as fava beans. So fava beans is actually the pictures on the left and the right. They are also called as broad beans. They can also be affected by certain infections and medications. And some patients may develop chronic hemolytic anemia. Some patients may be seen that some patients who have severe symptoms may develop chronic hemolytic anemia. The next one is the Duchenne muscular dystrophy. So in last seminar, we has discussed the Duchenne muscular dystrophy. So I'll just explain here. Muscular dystrophy is a collective group of inherited non-inflammatory but progressive muscle disorder without central or peripheral abnormality. So the picture below, so the Gower sign. So the Gower sign is actually they when this child starts to stand up, they will not directly use the leg. They instead will support themselves using their hands, and then they props their hands onto the knees to help them stand up. So these patients who have Duchenne muscular dystrophy can also have mental deficits and developmental delay, as well as progressive loss of ambulation. So by certain age, they will lose all they will have a loss of ambulations. Okay. Then the last one is the Weiling disorder. So Weiling disorder I put here is retinitis pigmentosa and hypertrichosis. So the first one is retinitis pigmentosa. So even the retinitis pigmentosa is related to Weiling, but they also multiple gene mutations that can happen. So for retinitis pigmentosa, the first symptoms that will appear for these patients is actually nyctalpia or night blindness, and they will get progressive blindness from these progressive blindness after they get the night blindness, but they can also see the photopsia or seeing flashes of the light. Okay. The last one is the hypertrichosis. For hypertrichosis, there are actually a lot of etiology of hypertrichosis. So for hypertrichosis, there is Weiling. Somehow I found that's actually located only in pinna or the ear. So the one shows in the pictures might not be associated with Weiling, but again, there are many etiology of hypertrichosis. So hypertrichosis is actually the growth of hair in excessive amount. The vellus hair will continue to grow up to 10 cm length in the entire body surface, except for the palm, soles, and dorsals, and the surface of recent phalanges and prefers. So the characteristics of this hypertrichosis actually, patients may also have dental abnormalities, defects in the ear, photophobia, and also physical and mental irritation depends on the severity of the disease. So then this is my references. So I guess that's all.
Any question? That picture of the Duchenne is among the commonest photo that comes out in OSCE as well lah. So you need to know about Duchenne. Duchenne muscular dystrophy is among the case that always come up in professional exam as well. You won't see the patient in the ward, you won't see the patient in clinics, but they will suddenly appear in your final professional exam. So read. I think other usual things other common things such as GCP deficiency are very common as well. Okay lah, alright, fine. One more.
Doctor.
If I may ask the presenter.
No, I'm not
Okay.
asking any question. I just like to ask you if I may add some info that I forgotten. So for achondroplasia just now, some of you might be asking why does only certain parts of the bodies are affected, as for the limbs only, not the head, not the ribs and such. Because of the receptor only responsible for chondroplastic sebab we have two types of ossification, intramembranous and also endochondral ossification, and only the long bones are elongating through endochondral ossification, but the bones such as our skulls and our clavicles are through intramembranous ossification. That's why only the long bones are affected, short, but not the skull, not the clavicles and the ribs. That's all.
Okay. Let's proceed. Last presentation.
Can you hear me? Hello?
Yes, I can hear you, but it's quite slow.
Okay. Bismillahirrahmanirrahim. Now, is it clear?
Yes, very clear.
Yes.
Okay. Okay, my name is Afika and my presentation would be on chromosomal disorder. So under chromosomal disorders or chromosomal abnormalities, it can be further divided into two, which are numerical abnormalities and structural abnormalities or defects. So first we go with numerical abnormalities. When we talk about numerical abnormalities, it means that there is gain or loss in the number of chromosomes. So actually under this category, they are further divided into two, which are first polyploidy. So, polyploidy and aneuploidy. Polyploidy means there is gain of one or more complete haploid chromosome set. Okay, so in us, in a normal human, we have total 23 pairs of chromosome, which is a 46 chromosome altogether. But in child with polyploidy karyotype, there is addition of another haploid chromosome set. So 46, like in a normal human, plus 23 because another set of haploid chromosome set, which results in 69 XXY as in a partial hydatiform mole. But I will not talk in details about polyploidy because our interest is to focus on aneuploidy. So all of these diseases, they fall under aneuploidy, which is more common. So what is aneuploidy? Aneuploidy is when there is gain or loss in the individual chromosome, not as a complete set of chromosome like polyploidy. So first we will go to Down syndrome. Just a brief introduction of this disease. It is the most common autosomal trisomy. So we are talking about autosomal chromosome, not a set chromosomes. So Down syndrome also considered as the most common genetic cause of severe learning difficulties. The incidence is 1 in 650 and the karyotypes are as follows. For clinical features, Down syndrome usually suspected at birth because of the baby's facial appearance. So as we can see in this picture, the affected infants, they are usually presented with hypotonic, which is low muscle tone throughout the body. The next picture is showing characteristic faces in Down syndrome. So this baby's posture is also due to hypotonic. And other clinical signs can also be presented, including flaccid occiput. This picture here shows a comparison with baby with normal head and baby Down syndrome with a flat occiput. So we can see the posterior part of the skull is quite flattened. Next is a single palmar creases. So in normal baby, also in us, we have 2 creases, but in baby with Down syndrome, they have only 1 creases. Incurved fifth finger, and a white sandal gap between the big and second toes. Okay, this is just the same as what I've mentioned. I'll provide it for your reference later. So aside from those that I've mentioned, these are the list of a typical craniofacial appearance, other anomalies, and later medical problems that can be presented with baby with Down syndrome. So this list I get from a Sunflower textbook. You can just simply refer to the textbook later. As for the cytogenetics of Down syndrome, we know that there is extra chromosome in chromosome 21. So actually there are different mechanisms on how there would be result in the addition in the chromosome 21. There are first mimetic non-disjunction, translocation, and also mosaicism. Okay, 94% of the cases is due to meiotic non-disjunction. Okay, what happened here is when there is error at meiosis, which result in the pair of chromosome 21 fail to separate. So one gamete has 2 chromosomes and the other one none. So when there is a fertilization of gametes with this unseparated chromosome, it will result in trisomy 21. So in translocation, 5% of the cases is caused by translocation, and the most common one is what we call as Robertsonian translocation. In Robertsonian translocation, there is extra chromosome that is joined together, that is joined onto another chromosome. So another way to put this is when these are one chromosome and this is the other chromosome, the long arm, and this one the short arm. What happened is when the long arm joined together and the short arm is lost. So in Robertsonian translocation, most of the time it involves only chromosome 14, but occasionally chromosome 15, 22, and 21 can also be affected. Another one is mosaicism. Mosaicism can happen in 1% of child with Down syndrome. In mosaicism, what happened is some of the cells are normal and some have trisomy 21. So how can this happen? It usually arises after the formation of chromosomal normal zygote. So when we look here, in mosaicism, the gametes are normal, but the problem arises after fertilization. So we can put mosaicism as an error in division after fertilization. These are all the explanation, same with what I've just mentioned. Okay, the risk of recurrence. In meiotic non-disjunction, it is related to maternal age. So after the mother having one child with trisomy 21, the risk of recurrence is 1 in 200 for mothers under 35 years old, and the risk remains similar after age of 35 years old. In translocation, the risk of recurrence is 10 to 15% if the mother is carrier and 2.5% if the father is the carrier. If the parent carries a rare translocation of 21 translocation, all of the offspring will have Down syndrome. If neither parent carries the translocation, which is most of the cases, risk of recurrence less than 1%. Mosaicism, if one child is Down syndrome, the subsequent chances is 1%. The risk factor for Down syndrome are older maternal age. So the table here shows the data of risk getting child with Down syndrome with maternal age at delivery. So as we can see, as the age increases, the chances or the risks are also getting higher. Next is related to family history. If the mother already have another child with Down syndrome, so there is subsequent 1% chance of getting Down syndrome baby. Also related to a carrier of genetic translocation. Here it shows the percentage of occurrence of getting child with Down syndrome. Carrier mother 10%, carrier father 25%. For the diagnosis, so we do ultrasound to look for nuchal thickening, which is the thickening of soft tissue at the back of the neck, and also we will look double bubble sign. The first picture here shows the comparison of nuchal thickness in normal fetus and fetus with Down syndrome. So this one is normal baby, this one is a baby with Down syndrome. So we can see the difference of thickness in the nuchal thickening in this ultrasound. The second picture here shows the double bubble signs that we can, which is expected to find in ultrasound. After that, when there is increased risk of Down syndrome, amniocentesis is also offered to check for fetal karyotype, and FISH, which is a fluorescent in situ hybridization technique, is also used. We also use a chorionic villus sampling to look for this finding. And in newborn, we take blood sample and do karyotyping and FISH. The first picture here shows amniocentesis, and this one is chorionic villus sampling. As for management, we need to convey this information and explanation to parents so that they will understand about the situation of their baby. So all of this is just quite straightforward. So maybe we can take some time for you to read. Okay, next is the recommendation for medical surveillance for children with Down syndrome. in this table, actually the components include, we need to monitor the complication in children with Down syndrome. There is thyroid test, growth monitoring, and so on. Other numerical abnormalities, which include first Edwards syndrome, trisomy 18. This is rare when we compare to Down syndrome. We can see the incidence is 1 in 8,000, as for Down syndrome, 1 in 650, which is more common in clinical setting. These are the clinical features that are expected to find in babies with Edwards syndrome. So this picture here shows the overlapping fingers. Next is Patau syndrome. Patau syndrome is also rare, even more rare compared to Edwards syndrome. You can see the incidence is 1 in 14,000, and the clinical features are as follows. Next, Turner syndrome. Okay, usually, more than 65% mother who have carries a fetus with Turner syndrome will result in early miscarriage. This can be detected by using ultrasound antenatally, where fetal edema of the neck, hands or feet, or cystic hygroma might be identified. In live-born females, the incidence is about 1 in 2,500, and here are the clinical features in children with Turner syndrome. So the child with Turner syndrome, we might also find short stature as the only clinical abnormality that we can find. The treatment, we monitor the height, cardiac evaluation, BP measurement, echo at baseline. We give growth hormone therapy, ovarian hormone replacement. Next is a Klinefelter syndrome, which occur in 1 to 2 per 1,000 live-born males. And the clinical features are as follows. This picture here shows the characteristic that we can find in children with Klinefelter syndromes. So these are all that are expected to be found. The treatment, we give androgen therapy and we treat the infertility. So moving on to the next category under chromosomal disorder, which is structural abnormality. Actually, there are many types of structural abnormalities, and Putri already mentioned about it in her part early on. But in my presentation, I will just talk about reciprocal translocation and deletion. So reciprocal translocation is meant by when there is exchange of material between two different chromosomes. So under this category, it can be further classified into two, which are balanced and unbalanced. Balance means when there is exchange in a chromosomal material, but the result there will be no loss of or gain of chromosomal material. So it's like it's balanced out. So when child that presents with this type of translocation, there usually will be no phenotypic effect on the child. And on the other hand, unbalanced reciprocal translocation contain an incorrect amount of chromosomal material after translocation occur. And usually this will result in both physical and cognitive development, leading to dysmorphic feature, congenital malformation, and also developmental delay and learning difficulties. The last one is deletion. So I mentioned I will just talk about reciprocal translocation and also deletion. So this one is an example of a deletion. Cry-Duchet syndrome. So Cry-Duchet syndrome is where there is loss or deletion of the tip of chromosome 5.
Cri-du-chat syndrome.
Cri-du-chat.
Cri-du-chat means cry of the cat. Cri, this is French. Cry of cat. I've seen one patient with this. I think while I was in UK. Very interesting.
Okay. So yes, like Dr. said, usually babies were presented with a high-pitched mewing cry in early infancy. Why they present with a mewing cry, high-pitched mewing cry? Because of structural abnormalities of the larynx and SNS dysfunction. So the severity varies. It depends upon the extent of deletion. Okay, that is all from me. Thank you.
Okay, any question? Clear? Faham? So chromosomal abnormalities, I think mainly all these 4 topics, usually it comes up in OSCE lah. But the cases that you may see in exams, in clinical exams, are Down syndrome. You'll definitely see Down syndrome in the exam. With Down syndrome, we have stocks of patients with Down syndrome. So Down syndrome, you can expect to come in exam. Duchenne, you will see in professional exam. Achondroplasia, I don't think we have any achondroplasia. We do have Edwards syndrome. I don't think they're gonna come for exam. We don't have any Turner syndrome yet. I think we don't have any Klinefelter. Yeah, basically Down syndrome lah. Down syndrome, Duchenne, those are the commonest that they would find and see in exams. But the others you need to know, it may come up usually in well, MCQ, anything can come out in MCQ, but OSCE, you'll definitely see all these sort of questions. Okay, all right. Someone said, Zakwan said it's a long night already, so I think he's asking us to stop now. Any question from others? Clear? Faham belaka? Faham tu?
Faham dah.
Okay, I think if everything is clear, means that, okay, again, I like to remind you, it's a very long way to go from exam. So read, and read. Okay, let's end our session with Tasbih Kafarah and Surah Al-Asr.